It started with a phone call that Frank, 68, would never forget. His brother David was on the other end of the line, calm but serious, telling him he had been diagnosed at stage 2. It was caught early, the doctor said, a lucky break. But what stayed with Frank wasn't the diagnosis itself. It was the strange way his own mind reacted in that moment. Instead of immediately thinking about his brother's condition, his first thought was something far more uncomfortable. He wondered when he had last had his own colonoscopy. That single thought quietly shifted something in him, turning a distant medical story into something personal. 3 weeks later, Frank was sitting in a gastroenterologist's office in Cleveland. His own results came back mostly reassuring. There was one small sessile serrated polyp, which the doctor removed on the spot. Nothing alarming, nothing urgent, just something to keep an eye on. The advice sounded simple and familiar. Adjust your diet, reduce red meat, increase fiber, and come back in 3 years. Frank accepted it the way most people do, as a routine instruction rather than a life changing warning. He went home and did what he was told. He bought psyllium husk fiber and mixed it into his morning orange juice. He added a salad to dinner, cut back on steak, and felt like he was taking control. For a while, it felt reassuring, like small actions could create safety. Then slowly the urgency faded. 6 months later, he barely thought about the polyp at all. Life moved on. The follow up appointment was still years away, and that distance made everything feel resolved. What his doctor never explained, not out of neglect but because science often lags behind clinical routine, was that eat more fiber is correct in principle but vague in practice. Not all fibers behave the same inside the human body. There is a specific category of carbohydrate, prepared in a very specific way, that changes what happens inside the colon at a biological level. It doesn't just support digestion, it actively reshapes the environment where disease risk develops. Researchers at Vanderbilt University uncovered this while trying to solve a paradox that had confused scientists for years, and what they found could change how that 3 year window is understood. Before going further, it's important to be clear about what this is and what it is not. This information comes from peer reviewed research, not from clinical advice. It is not a substitute for medical care. Anyone with a history of polyps, colorectal cancer, or digestive disease, or anyone under treatment should always work with a physician before making dietary changes. This is educational science, not personal instruction. The key discovery revolves around something called resistant starch. Despite the name, it is not an artificial supplement or rare compound. It is found in everyday foods like potatoes, rice, lentils, chickpeas, and even green bananas. What makes it powerful is not just what it is, but how it is prepared. The same potato, for example, can behave in two completely different ways depending on temperature. A freshly cooked hot potato acts very differently in the body than a cooled one from the fridge. When a potato is hot, its starch structure is gelatinized. This means digestive enzymes break it down quickly into glucose, causing a rise in blood sugar, and very little of it reaches the colon. But when that same potato is cooked and then cooled, something changes at a molecular level. The starch reorganizes into a form called retrograded resistant starch, or RS3. This form resists digestion in the small intestine and travels intact to the colon, where it becomes food for gut bacteria instead. This simple shift between hot and cold transforms the biological impact of the same food. A hot potato is quickly absorbed energy. A cooled potato becomes fuel for the microbiome. And this is where the research begins to get interesting, because what happens next is not just digestion, but a chain of microbial and chemical reactions that influence inflammation, cellular repair, and long-term colon health. And that is where the real story continues, moving from simple dietary advice into the deeper mechanisms that researchers have been uncovering step by step. Inside the colon, that undigested resistant starch becomes something far more important than simple fiber. It becomes fuel for a very specific group of gut bacteria that most people never think about. These bacteria ferment resistant starch and produce a compound called butyrate. And butyrate is not just another byproduct of digestion. It is one of the primary energy sources for the cells lining the colon. This detail changes the entire picture. The colon is not just a passive tube moving waste. It is a living tissue that depends on this compound to function properly. Healthy colon cells, called colonocytes, actually use butyrate as their preferred fuel, supplying most of their energy needs through it. When butyrate is present in sufficient amounts, these cells remain stable, efficient, and tightly regulated. When it is missing, the environment begins to shift in ways that affect inflammation, repair, and cellular control. The research becomes even more interesting when you look at how this system interacts with disease. Colon cancer cells, unlike healthy colonocytes, behave differently at the metabolic level. They rely heavily on glucose for energy, even when oxygen is available. This abnormal pattern, known as the Warburg effect, allows cancer cells to grow rapidly, but it also creates an unexpected vulnerability. Butyrate interacts with this difference in a surprising way. In healthy cells, it is fully consumed as fuel and never builds up. But in cancer cells, it cannot be processed efficiently. Instead, it accumulates. As levels rise, butyrate begins to enter the cell nucleus and interfere with enzymes called histone deacetylases, or HDACs. These enzymes normally silence certain genes by tightening DNA structure. When they are inhibited, the DNA becomes more open, and previously suppressed genes can become active again. In practical terms, this means that butyrate can help reactivate tumor suppressor genes and trigger programmed cell death in abnormal cells. The same molecule that nourishes healthy colon cells can, under the right conditions, signal damaged or cancerous cells to shut down. This dual effect is one of the most important discoveries in modern gut microbiome research. A study from the University of Michigan helped clarify how powerful this system can be. In a controlled trial involving healthy adults, participants were given a diet high in resistant starch, while others followed a control diet. Within just a few weeks, researchers observed clear shifts in the gut microbiome. Bacteria responsible for producing butyrate increased significantly, while markers associated with inflammation and disease risk began to decrease. These changes did not take months or years. They began within 3 to 6 weeks, showing how quickly the gut environment can respond to dietary input. But what stood out most was not just the speed of change, but the specificity. The diet did not randomly alter gut bacteria. It selectively amplified the very species responsible for producing protective compounds. This is where resistant starch begins to look less like a general dietary recommendation and more like a targeted biological tool. It doesn't just add bulk to digestion. It reshapes microbial populations in a way that directly influences butyrate production. And that production, in turn, influences inflammation, cellular repair, and the stability of the colon lining. At the same time, researchers began noticing something else. In populations with traditionally high intake of whole, unprocessed foods, particularly in rural regions where diets included day old grains, legumes, and cooled starches, rates of colorectal disease were significantly lower. These were not people taking supplements or following structured diets. They were simply consuming foods in their natural, often cooled or fermented states, unknowingly supporting the same biochemical pathway. What connects all of this is not a single food, but a metabolic chain reaction. Resistant starch reaches the colon intact, gut bacteria ferment it, butyrate is produced, colon cells are nourished, and at the same time, abnormal cells become more vulnerable to regulation and self destruction. But this system is delicate. It depends entirely on preparation, consistency, and the presence of the right microbial environment. Without those conditions, the same foods can lose much of their effect and the potential benefits remain unrealized. And as researchers continued to follow this chain deeper, they discovered that butyrate's role extends far beyond simple energy or even cancer protection, reaching into the very way genes are expressed inside colon cells. As researchers pushed further into this mechanism, they discovered that butyrate's influence goes beyond simple energy production or microbial balance. It reaches directly into how genes inside colon cells are switched on and off. This is where the science shifts from digestion into something closer to cellular programming. Inside every cell in the colon, DNA is wrapped around proteins called histones. Whether a gene is active or silent depends on how tightly this DNA is packed. When it is tightly coiled, genes are effectively hidden and cannot be read. When it is open, those genes can be expressed. This process is controlled in part by enzymes called histone deacetylases, or HDACs, which tighten DNA structure and silence specific genes. Cancer cells often take advantage of this system. They increase dull GDAC activity to silence tumor suppressor genes, the very genes responsible for slowing abnormal growth, repairing damage, and triggering cell death when necessary. In doing so, they effectively disable the body's internal safety system, allowing uncontrolled division to continue. Butyrate interrupts this process. It acts as a natural DDAC inhibitor. When OGDRDAC activity is blocked, DNA remains in a more open state. This allows previously silenced genes, including tumor suppressor genes like P53-related repair pathways, to become active again. In simple terms, butyrate helps restore access to the cell's built-in damage control system. What makes this more striking is how selective the effect is. Healthy colon cells use butyrate as a primary fuel source. They metabolize it efficiently and completely, meaning it never accumulates to high levels inside them. Cancer cells behave differently. Because of their altered metabolism, they cannot process butyrate effectively. Instead, it builds up inside them. Once concentrations rise high enough, butyrate moves into the nucleus of these abnormal cells and begins influencing gene expression at a deeper level. It does not act as a poison in the traditional sense. Instead, it changes the regulatory environment of the cell. In many cases, this shift activates pathways that lead to apoptosis, which is programmed cell death. In other words, damaged or malignant cells are encouraged to shut themselves down. This dual behavior is what makes butyrate so unique. It supports healthy cells while disrupting abnormal ones, using the same metabolic pathway. Researchers at Vanderbilt University demonstrated this effect in controlled laboratory studies, including experiments using germ-free mice. When butyrate-producing bacteria were introduced into these systems, the same selective effect on cancer cells reappeared, confirming that the microbiome itself was driving the mechanism. At this stage, the story is no longer just about diet or fiber intake. It becomes a question of metabolic design. The gut microbiome, when properly supported, is not just digesting food. It is producing chemical signals that influence gene expression, inflammation, and cellular survival decisions within the colon. This also explains why resistant starch matters more than generic fiber sources like psyllium husk in this specific pathway. Psyllium can improve bowel movement and support cardiovascular health, but it does not efficiently feed the bacterial species responsible for high levels of butyrate production. Resistant starch, on the other hand, is selectively fermented by those bacteria, directly amplifying this protective system. At the population level, studies have reinforced this pattern. Diets rich in whole, minimally processed foods, especially those containing legumes, cooled starches, and green plant foods, consistently correlate with lower rates of colorectal disease. This is not a coincidence of lifestyle alone, but a reflection of consistent biochemical input shaping long term cellular behavior. What emerges from all of this is a layered system. Food is not acting only as nutrition, it is acting as a signal. Resistant starch feeds bacteria. Bacteria produce butyrate. Butyrate reshapes gene expression, and gene expression determines whether cells remain stable or drift toward dysfunction over time. And as this chain continues, researchers began asking a deeper question. If butyrate can reprogram gene activity in colon cells, what else might it be influencing in long term tissue health, inflammation control, and disease prevention across the body? As this research expanded, scientists began looking beyond isolated cell behavior and focusing on what butyrate does across the entire colon environment over time. What they found was not limited to gene activity alone, but a broader restructuring of how the colon maintains itself day after day. One of the most important roles of butyrate is supporting the rapid renewal cycle of colon cells. The lining of the colon is replaced roughly every 3 to 5 days, making it one of the fastest regenerating tissues in the body. This constant turnover is not random. It is a protective system designed to remove damaged cells before they can accumulate mutations. When butyrate levels are sufficient, this renewal process operates efficiently. Old cells are shed, new cells replace them, and damaged genetic material is less likely to persist long enough to create long term risk. But when butyrate is low, this cycle slows down. Cells remain in place longer than they should, and the chance for mutations to accumulate increases. Butyrate also plays a critical role in maintaining the physical barrier of the colon lining. The cells in this layer are connected by tight junctions, which act like seals between them. These seals prevent harmful bacterial components and toxins from leaking into the bloodstream. When these junctions weaken, low grade inflammation can spread throughout the body, contributing not only to colon issues, but also to cardiovascular disease and broader metabolic dysfunction. This is where the research begins to connect gut health to systemic health. It is no longer just about digestion or even cancer risk. It becomes about inflammation control across multiple organ systems. A weakened colon barrier allows inflammatory signals to circulate more freely, which can influence aging processes and chronic disease development. At the same time, researchers observed that the composition of butyrate producing bacteria is not stable across a lifetime. In fact, it declines significantly with age, especially in populations consuming processed, low fiber diets. By around age 65, many individuals may have lost a large portion of their original butyrate producing microbial capacity. This decline is not sudden but gradual, shaped by decades of dietary exposure. Studies comparing different populations highlighted this clearly. In one analysis of elderly individuals, those consuming varied, whole food diets maintained a far healthier and less inflammatory gut microbiome compared to those in long term care settings eating more processed meals. Despite being the same age, their internal gut environments were dramatically different. This led researchers to describe resistant starch not as a supplement, but as a form of microbial refeeding. It restores fuel to bacterial populations that may have been depleted over time. However, this restoration is not instantaneous. It depends on whether the necessary bacteria are still present in sufficient numbers to respond. In some individuals, especially those with a long history of antibiotics or very low fiber intake, the response to resistant starch may be slower or weaker. In these cases, it can take several weeks for the microbial population to adjust, and in some situations, additional dietary support from fermented foods may be needed to help rebuild bacterial diversity. This variability is important because it explains why not everyone responds in the same way to identical dietary changes. The system is biological, not mechanical. It adapts based on existing conditions inside the gut environment. Even with these variations, the overall direction of the evidence remains consistent. When resistant starch intake is increased and when butyrate production rises, markers of inflammation tend to decrease and colon health indicators tend to improve over time. The effects are gradual but measurable. At this point, the focus of the research shifted toward a larger question. If dietary resistant starch can influence microbiome composition, genes expression, and cellular turnover in the colon, then how far reaching are these effects when maintained over months and years rather than weeks? As researchers followed these changes over longer periods, the focus shifted from short term microbial shifts to long term outcomes and disease risk. What became clear is that the colon is not responding to isolated meals, but to sustained patterns of feeding over months and years. Resistant starch, when consistently present, creates a steady environment that supports butyrate production and keeps the colon in a more stable biological state. One of the most important observations came from controlled dietary trials, where participants consumed high levels of resistant starch over several weeks. Within a short time, typically 3 to 6 weeks, researchers documented a measurable increase in butyrate producing bacteria. At the same time, inflammatory markers in the gut environment began to decline. These changes were not subtle laboratory findings, they reflected a clear shift in how the microbiome was functioning. But what stood out even more was what happened in longer studies involving larger populations. In extended trials, including long term follow ups after intervention periods ended, the protective effects did not simply disappear once the diet stopped. Instead, there were indications that some biological changes persisted, suggesting that butyrate exposure may influence longer lasting epigenetic patterns in colon cells. This is where the idea of durability becomes important. If gene expression in colon cells is repeatedly influenced over time by butyrate, then the system does not reset with a single meal or even a short dietary change. It adapts. Some genes related to inflammation control, cellular repair, and controlled cell death may remain more accessible even after the initial stimulus is reduced. At the same time, population level research has consistently shown strong correlations between high fiber, minimally processed diets and lower rates of colorectal disease. While correlation does not prove direct causation in every case, the consistency of this pattern across different populations strengthens the biological plausibility of the resistant starch and butyrate pathway as a contributing factor. One of the most cited examples in this area is a crossover dietary study comparing populations with very different traditional eating patterns. In these studies, participants from Western diets and rural, high fiber diets were temporarily switched to each other's food patterns. Within just 2 weeks, measurable changes appeared in colon tissue samples. Markers of inflammation, mucosal proliferation, and secondary bile acids shifted significantly depending on the diet being followed. In simple terms, the colon environment began to reflect the diet within days, not years, and these changes were not superficial. They reached down to the level of tissue activity, influencing how rapidly cells were dividing and how inflammatory compounds were being processed. Another key finding was that cancer related risk markers decreased in participants consuming high resistant starch diets, while they increased in those consuming more processed, low fiber diets. This pattern reinforced the idea that diet is not just influencing digestion, but actively shaping the biological conditions under which disease risk either increases or decreases. However, researchers also emphasize that this system is not uniform for everyone. A portion of participants in these studies showed weaker responses to resistant starch. This was often linked to differences in their existing gut microbiome composition. If key butyrate producing bacteria were already depleted, the fermentation process was less efficient, and the expected rise in butyrate levels was reduced. This helped explain why some individuals may need a gradual approach, starting with small amounts of resistant starch and potentially supporting the microbiome with fermented foods to rebuild bacterial diversity. Without that microbial foundation, the full biochemical pathway cannot function at its optimal level. Even with these limitations, the overall direction of the evidence remains consistent. Resistant starch supports butyrate production. Butyrate supports colon cell health, and colon cell health is closely linked to inflammation control and long term disease risk. As this understanding deepened, researchers began connecting these findings not only to colon biology, but to broader questions about how diet can influence long term genetic expression and disease prevention strategies across the human lifespan.